diagnosis and staging with Dr. Carrie Cheney, and then we'll take a break. So hang in there for the next little while, and I'm going to turn it over to Dr. Wood.
Thank you. All right. Would it be possible to dim the front lights a little? The slides will be clearer if that's technically doable. Can we do that? Can we dim the front lights? Oh, okay. Well, yeah, I'm just waiting for him to cue it up. Okay. Thank you very much for that introduction, Susan. As Susan said, I'm Gary Wood from the University of Wisconsin in Madison. And I have been focused on cutaneous lymphomas pretty much throughout my whole career. I started out in pathology and then moved into dermatology. And I just want to echo some of the things that Susan said. The Cutaneous Lymphoma Foundation is a wonderful organization, and everybody who is dealing with one or another of these diseases is very fortunate to have the organization in existence. A lot of people put in a lot of work for it, and it's a wonderful resource. So as Susan mentioned, I'm going to give an overview of cutaneous lymphoma, touch on a few things. I have no conflicts of interest. Then Dr. Carrie Cheney is going to talk about diagnosis and staging. There'll be a break. Then Dr. Bob Glinert will talk about the treatment of early stage disease. And then Dr. Stefan Schika will talk about the treatment of later stage disease. Then after that, we'll have a question and answer panel with all four of the MDs available. So the learning objectives for my talk are to give you a little background about basic immunology and skin biology so you can have a better, deeper understanding of what these lymphomas actually are. A little bit about their clinical appearance, although the other speakers will be getting into that in greater detail, and also just sort of an overview of how the lymphomas are classified. As Susan mentioned, the most common one is mycosis fungoides, which we usually abbreviate MF. And there's a leukemic version of that called Sézary syndrome, which often is abbreviated SS. And that's leukemic means that the cells, in addition to being in the skin or other tissues, they're also circulating freely in the blood. And that's much less common than the MF form of the disease. But we'll talk about that as well as other lymphomas. And then a little bit about how lymphomas develop and some general aspects of treatment, although the other speakers will be going into much greater detail about specific treatments. So first of all, what is a lymphoma? A lymphoma is a cancer of lymphocytes. Okay, that sounds good, but what are lymphocytes? Lymphocytes are a type of white blood cells, and there are three major types of lymphocytes: T cells, B cells, and natural killer cells, which are abbreviated NK cells usually. And they're derived from the bone marrow, and they mature in various places, the thymus, the lymph nodes, and the spleen. The T cells do a lot of their maturation in the thymus. What do these cells normally do? Everyone has these cells, and they're sort of involved in two types of immunity: adaptive immunity and innate immunity. And innate immunity is in evolution, this was the earlier, more primitive type of immunity. And this is where the NK cells come in. And they kill foreign or abnormal cells and help get rid of other threats to the body. But they do so with what are called invariant receptors on their surface. That is, all NK cells have the same molecules on their surface that they use to interact with other cells. In contrast, the cells that are involved in adaptive immunity have antigen-specific receptors on their surface, so they only respond to one particular type of substance. And they're divided up into two main types of T cells, helper T cells, which have a marker known as CD4, and they help other immune cells do their job. And then CD8 cytotoxic cells, which have the CD8 marker, and they kill foreign or abnormal cells. And then B cells, when they mature into their final stage called a plasma cell, they make antibodies and they secrete those into the circulation, and those are basically outside the cell, not attached to the cell, and they help fight off disease as well. So this is the origin of all lymphocytes. This is beef bone marrow. It's delicious. I don't know how many of you are fans of bone marrow, but I like it a lot. So schematically, in the marrow, all of the blood cells are derived from stem cells in the marrow. So the red cells that carry oxygen to your tissue, these cell fragments called platelets, which help your blood clot, for example, when you get a cut. And then these white blood cells, otherwise known as leukocytes. And there's two main types. There's myeloid cells, which are these here, and we're not going to talk about them today. And then there's lymphoid cells, which are the B cells, T cells, and NK cells, which are the ones that give rise to the lymphomas. So as I said before, after leaving the marrow, these lymphocytes of various types mature in the thymus, lymph nodes, and spleen. They, unlike other tissues where, lung tissue is just in the lung or colon tissue is just in the colon, et cetera, and when there's a cancer, they grow only in one spot until they get big enough to metastasize. These cells are normally, they're like commuters. They normally circulate throughout the body. They spend a lot of time in these tissues, the thymus, the lymph nodes, and the spleen, but they move throughout the body, and they travel, their highways are the blood vessels and the lymphatic vessels. And I'm sure you've all seen diagrams of the blood vessels coming out of the heart. These are the lymphatic channels, which is a sort of a simpler system, but it's an important system for the movement of lymphocytes, which, as I said, use both the blood vessels and the lymphatic vessels. And here is the thymus, which is up in the what's called the mediastinum, kind of near the heart, between the lungs, and the spleen is over here on the left side. And then there are lymph node groups, and many of you have had physical exams. Some of these lymph node groups can be felt during a physical exam, and that's one way we look for evidence that the skin lymphoma has spread from the skin to other places. So in terms of T cells in particular, which make up the bulk of skin lymphomas, there are different subsets of T cells that home to specific tissues like the skin, but there are other sets that home to the lungs or the gut or the joints, and these all have various markers and they all have different behaviors. So MF is a cancer of skin homing T cells as opposed to gut homing T cells. Now a little bit about skin structure. There's two main parts of the skin, the epidermis, which is the top part that you can just put your finger on right now while you're sitting there, and the dermis, which is underneath it, and then beneath that is the subcutaneous fat, which we're all usually thinking we need less of. And this is a very thin part. It's usually about 1 millimeter thick. This is the bulk of the skin itself, and anybody wearing a leather belt today, the leather belt that you are wearing is basically the dermis of cow skin. It's very rich in collagen protein, and it makes up the bulk of the skin, and it can vary in thickness. The back skin has some of the thickest dermis in the body. There are blood vessels and nerves, and there's appendages like sweat glands and hair follicles in the skin. And the different types of lymphomas tend to be centered, at least in their early stages, at different layers. So mycosis fungoides and Sézary syndrome tend to be centered up here in the epidermis and the upper dermis. Most other T and B cell lymphomas and advanced forms of MF as it grows and develops are centered more in the dermis. And then there's some rarer lymphomas that are actually begin in the subcutaneous fatty tissue here. And also, if any lymphoma gets big enough and aggressive enough, it will just enlarge so that it infiltrates the fat also. I'm showing here some T cells in the skin. And one thing to keep in mind is that there are about 2.5 billion T cells circulating in your blood at any one time in any person's blood. In contrast to that 2.5 billion, there are 20 billion T cells in your skin at any one time. That means 2.5 world populations of T cells in normal skin of an adult. And another way to put that is, if you look at your skin and think about 2 square inches of your skin, there's about 12 million T cells in that, which is greater than the metro populations of both Milwaukee and Chicago combined. So a lot of T cells in the skin. One of the terms that you may hear is the term immunophenotype. And all that is it's just a sort of a laundry list of markers, different molecules that are expressed either on the surface or within the cytoplasm that is internally in the tumor cells. And based on this, I'm not going to go into the detail on this, but based on a long list of various markers, we can tell what type of T cell we're dealing with. So in the case of mycosis fungoides in particular, it's a helper T cell because most cases have the CD4 marker, and it's a T cell because they express a thing called the T cell receptor, and it's a member of the skin-associated lymphoid tissue, which is abbreviated SALT. That is those cells that migrate to the skin selectively under normal circumstances, because it has certain markers such as CCR4, and that megamolizumab that Susan Thornton referred to earlier is actually an antibody that targets that. So the other T cells that go elsewhere that lack this are not affected by that antibody. It's not completely tumor specific, but it's tumor selective for MF. Now, the other thing that you may hear is you may hear about clonality testing. And clonality testing is something that, in the context I'm talking about, it is unique to T-cell and B-cell lymphomas because they have a tumor-specific fingerprint. And it's because they have on B cells here and T cells over here, they have what are called the B-cell receptor, which is essentially this is an antibody molecule, an immunoglobulin molecule that's just stuck to the surface of the cell. And as I said before, when these cells mature into things called plasma cells, they actually secrete these into the bloodstream. But in the B-cell stage, they just have them on their surface. And there's a similar type of molecule with a simpler structure on the T cells that's called the T-cell receptor. And the interesting thing about these is that although the parts of these are encoded by different gene segments, and these parts are, many of them are shared in common with other B cells and T cells, but when the gene segments come together that encode these proteins, there's the random insertion in the joining regions of other little pieces of DNA. And because it's random, it means that each B cell or T cell and then all of its descendants, sort of all the daughter cells, will have their own unique structure to this molecule. And because of that, it will, by chance, bind to some antigen in the environment. And then it will be a T cell that's specific for example, herpes virus or wart virus or staphylococcal bacteria, et cetera, any type of invader, or even foreign cells. Like if you have an organ transplant, like a kidney transplant, you have to be on immunosuppression because your T cells will recognize the donor tissue as foreign and will attack it if you're not immunosuppressed. And the same thing holds true for the B cells. So unlike a lot of other tumors, like lung cancer, colon cancer, prostate cancer, breast cancer, et cetera, they don't have these types of markers. These are only on these adaptive immune cells. And it gives us a tool to identify the overgrowth of one particular type of cell because they all have the same fingerprint, and we can detect that with various lab techniques. So now let's move on to what do skin lymphomas look like. Well, I'm going to talk a good bit about the T-cell lymphomas because they're the most common. Once again, mycosis fungoides and its leukemic variant Sézary syndrome. The earlier forms of MF are where people develop, mostly on sun-protected skin, patches which are completely flat areas on the skin, or plaques which are sort of like mesas in the desert. They're elevated a little, or they have some palpable depth. And that's the early phase of mycosis fungoides. It can progress to forming more like balls of tumor cells in the skin, and this is where the deeper dermis and even the fat can be involved. And sometimes you get ulceration or just erosion with these. And then sometimes the skin will be completely red, and that's classic for Sézary syndrome, but it can also occur in MF where you don't have any leukemic cells, but the skin is just diffusely red. And in Sézary syndrome, you have this red skin plus the circulating cells, and this is what they look like. These are the red blood cells. So these cells they're larger than normal lymphocytes, which are still bigger than red cells, but not this big. And they kind of have a very complex nucleus, which is the center of the cell. And to people that are used to looking at white blood cells in blood smears, they definitely stand out like a sore thumb as an abnormal cell. The second most common type of T-cell lymphoma that involves the skin are what are called CD30. So they have this marker called CD30 on their surface, and this is what that antibody brentuximab targets. There's sort of a spectrum here. So there's a spectrum that goes from clinically benign to clinically malignant, and pretty much everything in between. And the benign end is this thing called lymphomatoid papulosis, which is often abbreviated like this. And in this case, you get fairly small bumps that usually have destruction of the surface, so they end up being scabbed up spontaneously. And within several weeks, usually they will go away. But then more will come, and that cycle repeats itself. And this is a stain showing these CD30 cells. They have a mixture of some cells that are CD30 positive and some that aren't. At the other end are these anaplastic large cell lymphomas, which typically look like this, kind of a tumor nodule on the skin. And it can be up to several centimeters in size. And they are composed of more like sheets of these large cells, and there's widespread expression of this CD30 marker. And sometimes, even in the same picture, you can see some larger nodules and some smaller lesions. And there are people that have basically everything between this and this. And those are sort of intergrades here. And the whole thing is a spectrum, and it's very confusing to other dermatologists, even who don't see these diseases too often. And it's certainly confusing to most hematologists, oncologists, and other general doctors who also don't see much of these diseases. But the closer it is to this, the more benign the course is. Although this is, as you'll see in a minute or two, also a relatively favorable type of lymphoma. This is a type of lymphoma that is not favorable. This is called Berti's lymphoma, after Emilio Berti, an Italian dermatologist who first really described it. Primary cutaneous aggressive epidermotropic cytotoxic T-cell lymphoma, sometimes called CD8 T-cell lymphoma. And these form necrotic areas on the skin. They're very aggressive, and many people, despite everyone's best attempts, can go from being perfectly healthy to succumbing to this disease within a year. So how are the skin lymphomas classified? Well, we'll start with the T-cell lymphomas because they represent the lion's share, 75% of the total. And we sort of break them down into indolent, which have a 5-year survival of greater than 75%, many of them much greater than 75%. And the ones I have listed here This is mycosis fungoides and its variants, and we're not talking about Sézary syndrome here. We're talking about the non-leukemic forms of the disease. But those include a type where you lose pigment in your skin, hypopigmented. There's a type that clusters around the hair follicles called follicular, and there's another type that's in the skin fold areas called granulomatous slack skin. And there's some others too, but these are the main variants. And then this CD30 spectrum of lymphomatoid papulosis, anaplastic large cell lymphoma. So together, these make up 5/6 of all cutaneous T-cell lymphomas and about 2/3 of all skin lymphomas, including the B-cell lymphomas. So these are the most common ones that most people who get a skin lymphoma are in one of these categories. But there are some rarer ones. There's the subcutaneous paniculitic one, there's these other ones which we're not going to talk about here because we don't have enough time. And on the aggressive side of the cutaneous T-cell lymphomas, here we're talking about a 5-year survival of less than 25%, is the leukemic form of MF, the Sézary syndrome, also a natural killer type of lymphoma, this Birdie's lymphoma that I mentioned, a thing called gamma delta T-cell lymphoma, and this type of disease, which is prevalent mostly in Central America and Asia, but rare in the United States, as well as some unspecified types of lymphoma. And this type of lymphoma called adult T-cell leukemia lymphoma, which is prevalent, it's endemic in certain parts of southern Japan, and there is a virus associated with this disease that's also present in the Caribbean and some other pockets in the world. Interestingly, in the Caribbean, because the people there are immunogenotypically different, their immune systems are genetically different than the Japanese, this mostly causes a central nervous system degenerative disease in the Caribbean, whereas in Japan it causes the lymphoma. Same virus, but different outcomes. And you'll see that several of these, EBV is Epstein-Barr virus, several of these are associated with Epstein-Barr virus infection. There's also a neoplasm here that used to be called hematodermic lymphoma, but it's actually a different type of white blood cell of sorts that's called a plasmacytoid dendritic cell, but it usually gets lumped in with the lymphomas. On the B-cell side, we're talking about the minority of lymphomas. They're more common in Europe than in the United States, but in the United States, it seems to be about 25% of the total. And they're divided into indolent, which have a very good prognosis, 5-year survival greater than 95%. These marginal zone types actually have a 5-year survival of probably 100%. But these are the two most common, the marginal zone and the follicle center. And then there's another Epstein-Barr virus associated disorder called mucocutaneous ulcer, which forms on the skin and also mucous membranes like the oral cavity. And then there's some intermediate ones with a 5-year survival of like 50 to 70%, and that's this so-called large cell lymphoma leg type. There's an intravascular form of this lymphoma which forms right in the blood vessels. And then there's some other rare types. But once again, it's the indolent ones here that are the most common by far. So what causes lymphomas? The short answer is we don't really know the full story at this point, but it seems to be combinations of inflammation, infection, and mutations. And mutations are irreversible changes in the DNA of these lymphocytes. And these factors, what they kind of conspire to do is they stimulate cell growth, they inhibit cell death, which is a natural process in lymphocytes, and they select for the hardiest members of the herd. So this is kind of like a Darwinian survival thing. And so as these processes go on, you get more and more aggressive lymphomas. Selective growth leads to the emergence of dominant tumor clones, and because these are lymphomas made of T cells or B cells, we can identify those by the fact that the cell and all its descendant cells have identical antigen receptors, and there are ways in the laboratory to assess that. So conceptually, if you think about how lymphomas get going, you start with a normal T cell or B cell. There's a few different general pathways, a few different things that can happen. One is you can knock out genes that code for suppressor activities that sort of keep cells from growing out of control. And when that happens, you tend to have less what's called apoptosis. And apoptosis is programmed cell death. And this is an important part of sort of the balance in the body in managing white blood cells. So normally cells will be formed, and under various circumstances they'll die. And if you remove or block genes that mediate these processes, you'll get less death, and then you get what I like to call a lymphoaccumulative disorder. That is, it's not so much the cells are growing unchecked, they're just not dying when they should. They're like house guests that stay too long. On the other hand, you can have mutations or other changes, stimuli that turn on oncogenes. And oncogenes are genes that we all have that when they're abnormally expressed, really ramp up the growth and proliferation of these lymphocytes. And so you have increased proliferation. And these then are true lymphoproliferative disorders where the cells are accumulating not so much because they're not dying, but it's because they're growing really rapidly. And their doubling time, as we say, how long it takes to double the number of cells, is much shorter than normal. And you can even have situations where you go this route and then move over here. And this is my own personal view of what I think happens in mycosis fungoides. In the early stages of disease, it's more a situation of this, and later it becomes this. With some of the other lymphomas, they just go straight here. So just a quick example of that. So this C and D here, these are microscopic slides stained for a factor involved in the programmed cell death called FAS. And this is dermatitis. So this is a rash that's a non-malignant rash, just an inflammatory condition. And the T cells are expressing FAS. This is a case of mycosis fungoides with little balls of tumor cells up in the epidermis, the top of the skin. And you can see that the rest of the epidermis that makes up most of what you can see is expressing FAS, which is normal, but the tumor cells are not expressing it, which is abnormal. Somehow they've lost that. And down here, even when they haven't lost it, down here, these are various factors that are in that same FAS pathway. FAS is at the beginning of the pathway, and these are later. And these two are different types of things called caspases, which are proteins that have to be broken down or cleaved into smaller parts in order for the death sequence to occur. And in mycosis fungoides, in this particular case, although these factors are present in abundance, they're not being cleaved. There's just a few cells in which these two factors are being cleaved. And so again, the apoptosis, the cell death, has been retarded. Now, relatively recently, in the last several years, we have had what's called next generation sequencing technology, or high throughput sequencing. And that's allowed researchers to look at a patient's entire genetic composition and see what all their genes are doing and whether any of them are normal, and I shouldn't say most of them will be normal, but any of them are abnormal. And this is a summary that shows everything that you see here. The cell is obviously a very complex system, and these are all different factors that exist in your white blood cells. And all the ones shown in blue here have been found to, at least in some people with MF or Sézary syndrome, in the advanced stages. This is not early MF, but this is advanced MF. The ones in blue have either been amplified or mutated so that they're permanently turned on. And the ones in red have been either deleted or they've been mutated so that they're permanently turned off. And these are the various pathways here, and these are the various things that are turned on and things that are turned off. And here you see in the apoptosis pathway, this FAS protein is one that's been turned off. And one of the interesting things about mycosis fungoides and Sézary syndrome is that most types of cancer in their advanced stages just have a few key mutations. The typical case of advanced MF or Sézary has about a dozen of these mutations in the dominant tumor clone. And it's a little bit of a puzzle as to why there are so many mutations in these cells. But one of the clues may be is that the most common mutation that's present in more than 9 out of 10 people with advanced disease is this gene TP53, and it's shown here inside this little circle, TP53. It is involved with maintaining the integrity of the genome, maintaining the integrity of your DNA. And so it's possible that if that gets knocked out early on, it may cause a lack of surveillance that lets a lot of other mutations occur, and that's why so many of them can build up in advanced mycosis fungoides. Now, there's a few things that we don't know yet, which is how do these mutations actually evolve over time when you go from early disease to advanced disease? What's actually happening? How are they, what sequence are they acquired in? And also, how do they vary among the cells that are in the skin lesions? As they're evolving, are there different sort of subclones that all bear the same T-cell receptor, so you know they're from the original tumor clone, but they're picking up, you know, mutation number 1 over here, mutation number 2 over here, or is it mutation number 1, then mutation number 2 in the same subclone, and on and on. And this makes treatment of advanced disease challenging because there's lots of potential targets, and if you have a drug that targets one of these things, and there are drugs that target various parts of these pathways, that might be great, but it might not do anything for the other mutations. So what is the therapy strategy generally? You'll be hearing a lot more about details, but generally based on lymphoma biology. Well, first is I like to focus on what I call the 3 I's, which is itch, infection, and infrastructure. And so the reason to control itch should be obvious to anybody in the audience who's ever suffered from it. It has a lot to do with your quality of life. Infection is important to control, not only because if infection gets bad, it can go into your bloodstream, give you sepsis, and kill you, but also because there's recent, and I'll show you a reference in a minute, recent evidence from the Danish that if you treat, many people with MF have a staph infection on their skin. And if you treat the staph infection, you not only get rid of the infection, but you also reduce the proliferation of the tumor. There is some sort of driving factor that different microorganisms have on MF. And if you can treat people effectively, let me just see if I can go back here. Yeah, if you can treat the infection effectively, you can actually reduce the tumor burden that the patient has. So it's actually not only antibacterial, but also anti-tumor. And then infrastructure is the stuff that you all really have pretty much complete control over. So this is just general things that you do to support your immune system. With any cancer, if you pay attention to getting good nutrition, exercise, sleep, and mindfulness, and by this I mean trying to maintain a positive outlook, coping with reactive depression, which is very common in chronic diseases. If you pay attention to these things, all of these things have been shown to have tremendous impact on the health of your immune system, and you need the rest of your immune system to fight this or any other type of cancer. The good news is that many of the diseases, as we saw, tend to be relatively indolent, and they can often be controlled by relatively conservative skin-directed therapy that has low toxicity. However, in cases where there are multiple mutations, that can lead to treatment resistance because it's like playing whack-a-mole. You know, you hit one mutation with a drug, and then the others can pop up because they're not being affected. So aggressive disease often requires more toxic therapy, and typically combination therapy is better than using it's certainly better than just using one agent and then saying, okay, we gave it a try, and that's our best shot, and so that's the end of the treatment. But in between those is sequential therapy, where you use individual drugs one after another. And the problem with that is this issue of, with multiple mutations, you're going to knock out some of the cells with therapy number one, but then other cells are going to grow up, and then you're going to have a therapy number two. And none of the single therapies in general are going to be enough to wipe out all the tumor cells. And until you wipe them all out, they will come back. So there's less chance of treatment resistance if you use combination therapy. And many centers do use a lot of combination therapy, where somebody might be on 3, 4, even 5 agents simultaneously. Now, I just want to quickly end with some stuff that we've been working on over the last several years in my lab. And because of these things we've just been talking about with this tumor resistance, we try to develop novel therapies that kill cells faster by boosting apoptosis and slowing tumor growth so that the cells are killed before they can develop resistance. And there's several things. There's a drug called methotrexate, which has been around forever and is used for a lot of different things. And it turns out that it does a very nice complementary job when you use it with narrowband UVB. And we've done that clinically, and in the lab, when you use it with a type of light therapy called photodynamic therapy, it works very well. No clinical trials of this combo yet. And we're also working on methotrexate combined with a drug that inhibits the JAK-STAT pathway, which is one of those things in that complex mutation diagram that I showed you earlier that's wrong in MF. In addition, there's a purple dye called gentian violet, which is a very effective killer of MF in the lab, and we've used it on a few people because it's actually over the counter. You can buy it on Amazon. And it's basically used as a general antiseptic because it kills bacteria and fungi. And because it kills the bacteria, it gets into this paper by the Danish, Lindahl, 2019. Killing the staph reduces the MF tumor clone in the skin. So another potential reason to use this. The problem is that it stains your skin purple. And we're working on a method to change the color so that it'll be more cosmetically acceptable, but that we haven't achieved that reality yet. And then one thing I have illustrated over here is a combination of two drugs, a histone deacetylase inhibitor or HDAC inhibitor and a BET inhibitor. These BET inhibitor drugs, none of them are, at least the ones that we've been using, none of them are FDA approved for anything yet, but they are in clinical trials. This drug here, fedratinib, is FDA approved for certain bone marrow diseases. But the interesting thing about this combination is that it causes, this is a little heat map of all these different factors that help with apoptosis. And the red is where these genes have been turned on. And this is different time points. And you can see there's sort of a wave here. This is a 6-hour wave. And then there's a 48-hour wave where you got a few bright red ones. And then out here at 96 hours, you've got these ones down here get turned on. So there's like these 3 waves of apoptotic factors that are ginned up by this treatment. And the bottom line is that it ends up killing up to 90% of the CTCLs in the laboratory and also in Sézary bloods that we take out of the patient and treat in the laboratory. That's what ex vivo means. By 4 days, by 96 hours, 90% of the cells are killed. So that kind of quick wipeout is something that we're going for to try to kill these cells before they can mutate into something that's going to be harder to treat later. Now, there are no clinical trials of this particular combo yet, but the HDAC inhibitor we used is romidepsin, which is a FDA approved for CTCL already. A few other novel therapies that are under development. This one is some of my colleagues at the University of Wisconsin have made a radioactive drug here that has a completely novel targeting system. It does not target T cells in particular, but it targets all cancer cells because at least many cancer cells, different types. They have structures in their cell membrane called lipid rafts, and cancer tends to increase the density of these lipid rafts. This drug gets taken up in these lipid rafts, and then this radioactive moiety here will kill the cells that have taken this drug up. And in animal models of T-cell lymphoma, this has actually cured many animals that have T-cell lymphoma. So I'd keep my eye on this. It hasn't had any human testing yet, but it's a completely novel approach. Also, with the BET inhibitors, the group at Ohio State has been looking at their effect on what are called microRNAs, and there's this one microRNA 214, which seems to be something that when you block it, MF cells don't like that, and they tend to die. So they're pursuing the BET inhibitors from that angle. There's a German group that's published recently on inhibitors of BCL2, that's this agent, and this agent here, an inhibitor of NF-kappa B, and they have shown in the lab, again, not in human trials, but in the lab, that this is significantly active against cutaneous T-cell lymphoma. So, in summary, cutaneous lymphomas are tumors of white blood cells that home to the skin. The T-cell lymphomas are more common than the B-cell lymphomas, about 3/4 to 1/4. Mycosis fungoides and the CD30 positive disorders, lymphomatoid papulosis and anaplastic large cell lymphoma, together are the most common T-cell lymphomas, and fortunately, most cases are relatively indolent. Also, fortunately, most of the cutaneous B-cell lymphomas also tend to be indolent diseases. And indolent cases can be treated with low toxicity therapy in many cases. The aggressive cases do require more toxic therapy, but combination therapy often works better than single agent therapy for the reasons that we've already discussed.